Basically, if you look at the paper, we're gonna see how difficult the paper or how easy the paper was. Was it moderate, difficult, or was for at least everyone gave a chance to everyone to pass? So, if it was me writing this paper, these are some of the answers maybe I would have written if I was the one sitting in this exam. So, please take note that this is not the final memorandum, as the memorandum will be released officially. So let's go without wasting time. It is MSID, so that we're able to discuss about it. They're saying that change in the characteristic of species over time is known as what? So when you say change in the characteristic over time, you have the biological species, there's no change there. They're saying that population, this is a group of organisms these organisms which are similar, capable of interbreeding, produce a fertile offspring, living together in a particular time. So there is nothing related to change there. They're saying natural selection. This is not related also change in the characteristic over time. So if you look at evolution, remember evolution is a change, gradual change in the complexity of an organism. So since we are saying that characteristic of species, which is a living organism, therefore it is biological. So D looks to be the most correct answer, and you get the 2 marks there. Which one of the following option is correct combination of sources of variation? So we see that crossing over random arrangement of chromosome, crossing over correct random arrangement of chromosome, when? Okay, they have not specified. Mutation is also correct. But if you talk about mitosis, that makes it to be wrong. So we go to the next one, crossing over correct random arrangement of chromosome, correct. Continuous variation is not a factor which causes variation, but random fertilization is correct. So this one makes it to be wrong. We go to next one. They're saying random mating, correct. Random fertilization, correct. Mutation, correct. And meiosis, correct. Remember meiosis? We have not talked about random arrangement of chromosomes during metaphase, and we have not talked about crossing over. So those two are being generalized here. So C looks to be the most correct answer. They're saying that meiosis, mutation, discontinuous variations out. So C looks to be the most correct answer, and you get 2 marks there. They're saying that evolution involving long period of time during species undergoing little or no change followed by a short period of rapid. Guys, we discussed all of these things before you wrote the paper. Yes, we discussed everything. So let's just continue and find out where species undergo rapid change through natural selection. Lamarckism, no. Punctuated equilibrium, we say that is long period of no change and rapid period of a short period of rapid change. Long period of no change, rapid short period of rapid change. So it keeps on going like that, like that. So it's like a stair. So this looks to be the most correct answer. Artificial selection, not really. Gradualism, you see it is saying it's gradual, so it's out. So B looks to be the most correct answer. So we go to the next question, 1.1.4. Which one of the following fossil does not belong to Australopithecus africanus? Mrs. Place is correct. Lucy is Australopithecus afarensis, Littlefoot and home child. So B looks to be the most correct answer in this case. You get your 2 marks. 1.5, which one of the following is only found in RNA nucleotide? RNA nucleotide, when you say RNA nucleotide, here we are looking at adenine nitrogenous bases. We are finding it here. The key point here is only and RNA nucleotide. So they're saying uracil nitrogenous base, correct. This one, adenine, we find it in RNA and DNA, so it's not, so it makes this one to be wrong. DNA, deoxyribose, only found in DNA. Phosphate, we can find it in RNA and DNA, so it's out because of the word only. So B looks to be the most correct. We go to the next question. You're saying that 1.6 to 1.7 are based on the diagram below. Okay, you have a black Henny, I don't know whether it's a hen or a hawk, and then a white one, then you cross them, you get gray. So you're getting an intermediate. So that's the key point there, that you are getting an intermediate. So we are saying that which type of dominance result in intermediate phenotype that's shown in the diagram above. So that is incomplete dominancy. Ne? Yes, it is incomplete dominancy. So the answer becomes C, because codominancy, you have a mixture of the two. Yes. So if you look at this, it looks to be a mixture of black and white, but that's as a result of not the perfect picture. But since they specified it here and they said gray, so it makes it to be more sensible. So C becomes the most correct answer. 1.7, the genotype of gray of chicken is, so since it is a combination of the two, the combination of the what? Of the two, so it's going to become capital B, small b, capital W. Why do we say that? Because of the B, capital B is here, and then you have a capital W, so that when you mix them together, you form capital B, capital W, which is incomplete dominancy. However, even the codominancy is the same type of genotype. We go to 1.8. The diagram below shows the formation of the sperm cells of fruit fly. Okay, sperm cells of what? Fruit fly. Fruit fly, this is what called a Drosophila melanogaster. Yes, scientifically we call it what? Drosophila melanogaster. So this Drosophila melanogaster, we're saying that it has 20 chromosomes in the nucleus, and then you have after mitosis, meaning that the number of chromosomes remain the same. The number of chromosomes remain the same. But now what happens? After meiosis. You see, this is after meiosis, and meaning that now you have produced the what? The gametes. So it means that this becomes 1N. Into N, it's 1N. How many chromosomes are present in W and D? So it means that we're going to find out that in W, we're going to have W, this one, we say it's 2N, meaning that is 20. And then this one must be 1N, which means it is 10. So we have to look for that, which is this and this. So D looks to be the most correct answer. Then we go to the next question, 1.9. The diagram below represents the different phases of meiosis, saying that which one of the following correct sequence of the phase. So we see that X, Y, Z. So you see that the correct, which one of the following represents the correct sequence of the what? The correct sequence of the, so we see that this is metaphase. See, they're in the middle, yes, and then this is anaphase 1. Why? Because they are still chromosomes. Well, this is anaphase 2. You understand? So because the anaphase 2, they are chromatids. You see, they are chromatids. So it means that I'm going to start with 1. I go to 2. I go to 3. So Y, it must start with Y. So whatever is not Y is out. So it's going to be either this or this. So we go to the next one, which is 2, which is X. So it's going to be YX. This is YZ. So this one out. So this one looks to be the most correct answer. And then you see that is Z. So automatically that makes it to be the most correct answer. Then we go to question 1.1.10. The diagram below shows the DNA molecule on the left and a pool of few nucleotides on the right. Original. So you have the original DNA molecule, then the new DNA molecule. If replication of original DNA molecule takes place using new nucleotides, what would the product of replication look like? They say they are using this. So it means that the original must look like this, and then the new must look like that. You understand? So it means that, remember, in the DNA replication, this structure is supposed to unwind so that each is forming a template. You understand? So it means that this one must be on one side, must be on one side. Also, this one must be on the other side. Then it opens. You understand? So even this one must be like that. So let me just use a different color so that now this one is the red one. So then you're gonna have to be with another strand here, form another structure, and then also another strand here to form another strand, so that now it means that this the red one becomes the new, and then this blue one becomes the old. So we have to look for something like that. So we see that this one is out, this one is all new. Then you see it has the old one, which is the blue, and then the black one also, which is the blue, and then you have the red one, which is this one, which is the new one, and then this one, which is the new one, because this and this, they became template for formation of the new strand. So C looks to be the most correct answer. And then I will say that C, you get 2 marks, and this marks our objectives, who got 20 out of 20. And if you got that, congratulations for this achievement. Then we go to the terminologies. We are saying the diagram below used to show the evolution relationship between the species. How do we call that diagram? We call it phylogenetic tree. Then we are saying that ability of chimpanzee to walk on 4 limbs. So we say that is quadrupedalism. So we get a mark there. The part of the skull where that houses the brain, they brought this one even in paper 1. So it is cranium. They are saying the structure in the animal cell that give rise to the spindle fibers or spindle threads. We call it centrioles. So those are 2 marks. Sorry, 1 mark. Saying that sex-linked genetic disorder where blood fails to clot or does not clot. So we call it hemophilia. So also another mark. They're saying that the genetic state, the genetic state of having 2 identical alleles for a particular characteristic. So how do we call that? Genetic disorder having 2 identical. That is homozygous. So it becomes homozygous. Then they are saying that an organelle containing DNA found in plant cell and not in animal cell. How do you call that organelle which has DNA found in plant cell only, but you cannot find it in animal cell? So we say that is a chloroplast. Yes. So that is it, guys. I hope you got all the 7 out of 7. If you got 7 out of 7, congratulations. If you didn't get that, 5, 6, still is fine. All right, let's go to law of use and disuse. Who said that? Basically, it is Lamarck who said that. So the answer is B, only. The word only must be there. I've been emphasizing that. They're saying that because this one they talked about punctuated equilibrium. So they're saying that chromosome complete complement of women with Down syndrome. Remember, Down syndrome is chromosome number 21. But here they're saying 44. So since they're saying 44, they are supposed to be 45. And then XX, yes, but since this is 44, so this is a normal person. And then this one, it is not XXX because Down syndrome doesn't happen on a sexy chromosome. So the answer looks to be none. Then they're saying that the father of genetics, we call it Mendel. Mendel is the father of genetics. So we get also there 2 marks. So it is A only. So basically it is B only, none, and then A only in that regard. So I hope that one was also a little bit simpler. You got 10 out of 10. Then we go to the next question. They're saying that Mr. Forbes has had a baked graduation cake for her sister and then left in the container in the cupboard. When she returned from work, all remained in the container were crammed from the cake and trace of blood droplets where the thief had grazed his or her finger on the sharp part of the container. It's a DNA sample of her two children, along with the DNA sample from the blood found in the container. All right, DNA sample of child A and DNA sample of child B. So they're saying that name the technique used to compare these results. Remember the definition, they're no longer asking it, but now they're asking what is this technique. So we say that it's called the DNA profiling. There's a difference between DNA profile and DNA profiling. DNA profiling is the technique, while DNA profile is this what you get after using the what? After using the technique. So give the letter of the child who most likely ate the cake. So in this case, we're gonna have to look at, because they say that this goes with that, goes with that, this goes with that, this goes with that, this goes with that. So it looks to be child number B, child B. Then they're saying that from the list below, write down the letter of two correct uses of the technique mentioned above. So they're saying that from the list below, write down the letter of two correct uses of the technique. Use. Oh, they gave you what is here. So basically, cloning, no. Identification of genetic disorder, correct. Establishment of family, what? Family relations, yes. Genetic engineering, no. So the answer is B and C. Yeah, B and C. Yeah, I see, guys, the paper is still moving well. It's not that bad. It's not that bad. At least it could be a recovery from paper one for those who are crying for paper one. How do you see? Tell me in the comment section, and then I find out how do you see this paper, or how do you see this paper was. They're saying that diagrams A and B show the skull of two hominids. Skull of two hominids, and then you have skull A, and then you have skull B. So if you're saying that you have skull A and skull B, large cranium. So where is the large cranium? Which diagram, or if you look at it, you see that here it has a big space of the what? Of the skull. So it looks that B has the biggest cranium. Skull of a chimpanzee, the skull of a chimpanzee. Chimpanzee walks almost on two limbs, so the foramen magnum must be in a more backward position. So B, and also if you look at the teeth and the shape of the palate, you see that is rectangular. So I will say that skull A is Z for chimpanzee. Then they're saying that name the opening to which the arrow are pointing. So how do you call that opening? The opening is called foramen magnum. Foramen magnum. Yes, so that's what we call. State the shape of the jaw. How do you call that shape of the jaw of A? So you see that we say that is a rectangular. It's a rectangular shape. Yes, and then they're saying that give a letter of the diagram A and B, A or B, representing the bipedal hominid. Bipedal, why do we have to say A or B? In this case, I will say B. Why? Because of the position of foramen magnum is in a more forward position. If you look, yes, it is in a more forward position. But they did ask you to give a reason, so I will use that to identify as. We shall say that this is B. They're saying which skull one or two better adapted to chew raw uncooked food. I like the way they have asked these questions. Yes, because we have been asking them the other way around. Yes, the person who said this paper, yeah, you are good. So now we are saying that I will say in this case, I will say that which skull in diagram is better adapted for chewing raw uncooked food. So I will say in this case, I will say A. Why am I saying A? Because of the teeth, the kind of teeth this species is having. Look at the kind of teeth this species is having. It shows that it was feeding on raw tough food. So because he was feeding on raw tough food, so I will say that that is the answer in that regard. So give the scientific name of modern humans. Remember, I told you that whenever you are writing the scientific name, you have to write the first name, which I call the surname, which I call in science the genus name. The genus name must start with a capital letter, and the species name, or what I can call your names, it must start with a small letter. You don't stop there, you underline. Homo sapiens. Homo sapiens. So, since you are not a computer, you can't write it in italics. So it is Homo, yes, not Homo. Homo, then sapiens. Yes. So after that, you underline. Make sure that you underline. But make sure that this letter is a capital letter, and make sure that this one is a small letter. If you see that mine is big, then you can write it small. Sa Homo sapiens. Yes. So please, 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 you must always write it like that. Yeah, you see, this question was not that bad. You could get all the 7 marks here. Yes. Okay. We got to question 1.6. Let's go to question 1.6. We are saying that, what are they saying in question 1.6? They're saying that in humans, one gene controls hairline and another controls freckles. The hairline can be curved, forming a point down, meaning this, pointing down the forehead, called Windows what? Peak, H, capital H. And then that is straight like this. I think it is small. I think even this one has the same thing. Freckles, which are tiny brown spots on the face of the what? Present, these ones. They're saying that the diagram below shows a cross between a male and a female who are heterozygous for both genes, meaning that this is heterozygous, okay? It's heterozygous, and this is heterozygous. And then if you look at this, we shall see that capital F, small f, when I do the gametes, this gonna go with this, is gonna be capital H, capital F, then this is gonna go with this, capital H, small f, and then this with this is gonna be small h, and capital F, and then this with this is gonna be small f, sorry, small h, and then small f. All right, let's see, confirm, and then it's even the same story here. It's gonna be the same. So if I have these ones, correct, I have it, correct. And I have it, Hmm? What's happening here? Hmm? Is there... this looks to be a small mistake. I don't know how they're gonna mark it, but basically when you look at it, it is supposed to be small h, small f, because if you have these gametes, so these gametes, they came, these gametes are supposed to be transferred here. You understand? Yeah, the same way these ones, because it is the same genotype, this one has this, which are not resembling this. So there was a small mistake there. But let me see if them writing it like this will influence your answers. Yes. All right. Name the type of genetic cross represented above. It is a dihybrid cross. Okay, correct. State the dominant characteristic of hairline. Dominant characteristic of hairline. They said it here. It is the Windows peak. This one, Windows peak, they said capital H. Windows peak hairline. All right. So we have not yet influenced that. Name the process in the reproduction mentioned above. Name the process in reproduction indicated by above. Remember, this is basically showing us that, oh, it could be fertilization, because this is fertilization, but also this one should be this to form the gametes. It is meiosis. You understand? And then Still, I can also see, because I see a sperm here, so it can also be gametogenesis. All right. Because I'm seeing a sperm here and seeing a sperm. So it can't be oogenesis and oogenesis and gametogenesis. Sorry, and spermatogenesis. So I'll maybe have combined it and then I say gametogenesis. Or how do gametes form? They form through meiosis. All right. Let's find out what they're saying. Arrow. Where is the arrow? Arrow. Oh, these arrows. What am I forming here? So basically, it looks to be much more accepted as gametogenesis. And then they are saying that Pinnet square. The Pinnet square is basically fertilization. Remember that we always write here that fertilization. So that is it, fertilization. They're saying that the genotype of offspring X, so now this is going to affect us. The genotype of, so I'm going to continue the way it is there. I'm going to say capital H, capital H, capital H, capital H, small f. Oh, wait. I need to see properly. It's going to be this with this. So it's going to be capital H, capital F, small f. And then this one is going to be capital H, small f, small f. You see? Yes. But if someone had written, if we were supposed to take it the way it was supposed to be, remember, it's supposed small h, so it would have been capital H, small h, capital F, small f. So instead of capital H here, it would have been small h. If we have taken it, if like the proper way, the way how it's supposed to be. But it will depend on how they have marked. But obviously, this, if we continue the way it is here, if you did not maybe see it, so it could be the mark there. Yes. So basically, it is gonna be like that. So just cross this with this so that I'm able to get this. And then to get this, I have to cross this and then with this. So where they meet, and then that is why. Or the allele for the start, as long as it is allele of the same character. So if you look at this, capital small h and then capital F, because these are two different alleles. That's why F does not influence the existence of H. All right, we go to the next one. Give the genotype of this, so we have seen it, and then give the phenotype of this. The phenotype of this, so it is going to be affected again based on this caliber we have talked about. It's going to be is capital H, small h, small f, small f. So it's gonna be what? It's gonna be capital H is window. That is capital H is gonna be windows, peak, and small f. Small f is what? Small f, small f. Wrinkles which are brown, tiny brown spot, then to f. So it's gonna be and then the other one is absent. So basically it's gonna be because this one small f means wrinkles are absent. So wrinkles absent. So it's gonna be capital H, which is Windows speak, Windows peak hairline, basically with the wrinkles being absent. Yes, that is if I follow this. But now, if it was supposed, if it is going to be marked as the way it's supposed to gonna be, this is small h, so it's gonna be the other one is what? straight hair and straight hair and hairline, and then freckles being absent or absent of freckles. Yeah, so that question will depend on how they have discussed it in the memorandum, but that is the concept behind. I've talked about human, then it means that whatever you are writing, it must be linked to 46 chromosomes or linked to human. The cell ABC shows different events in the phase of meiosis. So basically you have A, you have B, and you have C. So remember this process which you see here, it's crossing over. And then you need to know what happens during the crossing over when the chromatid of one adjacent chromatid of one homologous chromosome cross over with another chromatid, and then at a point called chiasma. And then because of the variation we see here, we see that the crossing over took place on top of the two adjacent chromatids of the different chromosome. So now they're saying that provide labels for structures W. No. Y. Where is Y? Y, it is a cell membrane. Why? Because it's a cell. They said it's a cell, so it is a cell membrane. The nuclear membrane has started already to disappear, so we cannot include it here. And then what is Z? Z is the centromere. Centromere. So we got the next question. Name and describe the process that caused the chromosome in C to look different from the original cell in A. What caused this to be looking different from this? So in this case, the process, they said name the process. So crossing over, and then now you describe what is being involved. Like basically you tell us what happened because they say that question 2.1.24 here, no. Name and describe a process that caused the chromosome in C to look different from the original chromosome in A. So what caused that? It is basically crossing over. Chromosomes they line together, or homologous chromosome align together or next to each other. Chromatids. Now you have to talk about each chromatid. Each chromatid of maternal and the paternal chromosome overlap. But it's not basically better to say each chromatid, because if you are looking at this, because we're explaining this, adjacent chromatid from the paternal and maternal, yes, overlap at a point called chiasma, and then genetic material is being exchanged. So in this case, basically they are looking at process of crossing over. But in this question, there is a bit of application. They want you to show us that it is this and this which crossed over. So these are adjacent chromatid, one from the father, one from the mother. Yes, you understand? They must be adjacent because you have also this and this. So adjacent chromatid overlap at a point called chiasma, and then they exchange the what? The genetic material. Yes, so basically that's what you're supposed to explain, because you're supposed to explain based on what you see there. Then we go to the next question. Look, this answer was not supposed to be, this answer is supposed to be there. They're saying that explain the impact of process mentioned above for survival. So they want you to tell us that what is the function of this crossing over in terms of the survival of the species. So basically it leads to variation. That's number one. It leads to variation, yes, in offsprings. And then if there is variation in offspring, we're going to see that the organisms are going to survive differently. So it leads in variation in offsprings. Yes. The species will be able to survive, to become better. They have better chances of them to survive. If there is variation, it will lead to better chances of what? Of survival. And then also, I always ask you a question that some of us, we might not look like our fathers or our mothers. What causes that? It's because of the genetic variation. So it will result, it means that each gamete is totally different from one another. So it will lead to difference in the gametes genetically. So it will lead to genetic difference, you understand, or genetic variation in gametes. So you can also talk about that or any other which is proven to be correct and it's related to the variation which is being asked. Draw the diagram to show the chromosome that would appear in B during the process named above. Which 1.1. Okay, it means that they want us to draw what would have happened here. So in this case, I will say that you remember that whenever you are drawing, you have to put a caption. But now, when you are drawing here, we want to see that this chromosome is like this, is white, or it has a chromatid. You have to draw it properly, ne? It has, let me just draw it like this, guys, ne? Just to show you properly here. And then this one is black, you see? And then this is black. Then you shade them. You understand? You shade them. So this is black. So that this part cuts off and joins here. And then the white one cuts off and then joins there. This one joins here, you see? So it means that it is this chromatid and this chromatid which overlapped. And where is the overlapping happening? It is happening up. It's not happening down. So basically, that is what you're supposed to draw. And then you put your caption here. You don't forget to label. So this is the chiasma. And then you tell us this is... you draw actually you are drawing this inside this cell. You tell us this is the cell membrane, this is the cytoplasm, this is the chromatid. So you tell us this is a homologous chromosome under crossing over, or sometimes homologous chromosome under crossing over, which we call bivalent. Yes. Then we go to the question. I hope you got the 14 out of 14 there. The diagram below represents the process that occurred during protein synthesis. So you have the molecule, okay? And then now protein synthesis, molecule B. So we see that here you have this process. Molecule Y, we say is a ribosome. Since this one is forming, so automatically it means that molecule B is going to be transcription. All right. Now, let's find out what does it mean. Identify molecule A. So we see that in molecule A, where is molecule A? Molecule A is DNA. We see that that is DNA molecule. Then we have molecule B. It is they are looking for this. So it is RNA molecule or messenger RNA. We can say messenger RNA or mRNA. Describe 3 structure features of molecule A. Molecule A. Remember when I was asking you before you went to the exam, I said that please don't go to the paper without knowing to describe the structure of DNA. So they're asking you, describe the structure features of that. So you tell us that is double helix. DNA is double helix, or molecule A is double helix. We know that's double helix, or it's also double stranded. Remember, double helix means that is helical like this. Yes, also this helical, so it becomes double helix, but still it has 2 strands. So it's double helix. You can say it's double helix. You can say it's double stranded. You can talk about it's made up of nucleotides which have deoxyribose sugar and a phosphate. You can talk about it has adenine, thymine, guanine, and cytosine. You can talk about it has thymine. Basically, it doesn't have the uracil. So don't talk about the uracil because they're asking about the structure. What are the monomers called? It's monomer. You can talk about, sometimes you can say monomers or building blocks. It's made up of double helix. So basically you explain that. Yes. So we go to the next question. And then what are they saying? What are they saying? The structure function of Y. What is Y? We come back and then see, oh, what is Y? So Y is a ribosome. And we said that the function of Y is to allow this messenger RNA to be carried, to give a platform for the molecule, for the molecule messenger RNA to allow translation to take place. So basically, it is a site for protein synthesis. It's a site for protein synthesis. Name and describe the stage of protein synthesis that takes place in Y. When I was discussing about this yesterday, someone was asking me, sir, why are you describing only translation? Then I told them, make sure that you know it before you go to the paper. Translation. I talk about transcription. No, I didn't talk about transcription. I didn't describe transcription, but I was very clear about translation. Yes, because basically you need to listen to your teachers. Yes. All right. You're saying that describe the process of translation. There we go. Describe for me the process of translation. Basically, that's what they are looking. So you describe according to the messenger RNA codon, the transfer RNA with the complementary anticodon, it brings the required amino acid, the required or specific amino acid to the ribosome. The amino acid are joined together by peptide bond to form a required protein. So you're supposed to explain the process of translation, or you describe it according to what your book is saying, as long as you talk about all the codons, the anticodons, the amino acid which is required, or specific amino acid to the ribosome. You describe required protein, the peptide bond which joins the two amino acids, the different amino acids together. So you're supposed to describe that. Yes. We got question 2.3. 2.3. Let's go to question 2.3. What are they saying? They are saying the information below shows the sequence of protein in the first 6 amino acid found in the saliva of Angela and Brian. The mutation caused by change in the Brian sequence of amino acids. The amino acids are caused from left to right. They're saying describe how mutation caused the change. Describe how mutation caused the change of the Brian's amino acid sequence. Describe how a mutation caused the change to Brian's amino acid sequence. So basically, how mutation is caused by change in the sequence of nitrogenous bases in the DNA. Basically, the moment it's a mutation, which is a gene mutation, it is caused as a result of the change in the sequence of the nitrogenous bases in the DNA. So this change will lead to the change in the messenger RNA sequence. So remember that you have DNA, you have messenger. So if the sequence of DNA is changed, it will lead to the change in the sequence of the what? Of the messenger. And then this in turn will change the sequence of transfer RNA. This will change the sequence of transfer RNA, that is anticodons. And then this will change. If you look at this sharp, check the fourth amino acid. The fourth amino acid, but this one is fine. The fourth amino acid, yes, of Brian will be changed. You understand? Yes, will be changed, because we see that the Brian is valine, while Angela is proline. So it means that instead of the proline, then valine is gonna be given out, meaning that now, since there is a change in the amino acid,
yes, then automatically the protein is going to what? To change. So basically, you tell us that a change in the DNA will result in the sequence of DNA bases will result in the change in the sequence of messenger RNA, which will result in the sequence of transfer RNA, and then this will lead to the change in 1, 2, 3, 4, the fourth amino acid from being proline to valine, and then the protein definitely will have to change. They're saying how many messenger RNA nucleotide would involve in the portion of protein shown in Angela's. Okay, they're saying it's 1, is 2, is 3, is 4, is 5, it's 6. So 6 times 3 is 6 times 2 is 12, is 18. Remember why 18? Because they're saying that there's how many messenger RNA nucleotide. They're saying nucleotide. Nucleotide is equivalent to the base. You understand? Yes. It's equivalent to the what? The base. Because remember, each codon on the messenger RNA is coded by 3 bases or 3 nucleotides. So if I have 1, 2, 3, 4, 5, 6 nucleotide times 3, 6 amino acids times 3, because each amino acid is made up of 3 nucleotides, you understand, or 3 bases. So automatically it gives me 18. They're saying write down the DNA base triplet from left to right for messenger RNA sequence above. From left to right for messenger RNA sequence above. So I see that is it G? Is C, is U, is G, ne? So I'm gonna say it's gonna be G, it's gonna be A, and it's gonna be C. And then this one is gonna be T, it's gonna be G, it's gonna be C. Why? Because they're saying write down the DNA triplet sequence from left to right of messenger. So if this is messenger, automatically DNA has thymine, doesn't have uracil. So I can't put uracil here. So it is thymine. So it is this, and then you get a mark there and you get a mark there. Who got 7 out of 7 in that regard? I see that things are moving well when I check here in the chat. I see Zoo, you guys, I Kamahero, I see Speciation, but I see you guys. Someone is specifically asking me about the other question that if I don't underline. Yes, if you don't underline, they might give you, but make sure that you underline. You must always, but if you don't put a capital letters, you are gone. You are gone. So that is it. So let's go to the next question, which is question 2.4. Okay, question 2.4. Uh, 2.4. Okay, question 2.4. All right. So we are saying just a moment, guys. No, today's network. Okay. All right. Oh, it's this one. All right. So we were saying that. Recording in progress. All right. Okay. Yeah, I'm back here. We have been having challenge. All right, we are saying that the next question is saying that if you are saying that, so what we are saying that the graph below shows the percentage. Let me just clear up and then I repeat the question. I don't know whether you got it before this technology is disturbing me. All right, they're saying the graph below shows the percentage distribution of the world's population according to the blood group A, B, and O. Guys, by the way, let me know if you are hearing me from the what from the chat, because you know sometimes things are not. Whether please type and say that yes, we hear you. Whether they have a positive or a negative form. Okay, they're saying that the percentage distribution of the world's population by blood group, saying that according to the data, yes. Or sorry guys, I thought that maybe you didn't see it, so let me just restart this question again. All right, they're saying that according to the data, what is the total percentage of the world's population of blood group O? So we said that this blood group O, we say that it is this, and no, it's this and this, which is 42 plus 3, which gives me 45. Yes, and then we said that the tabulate, you must draw a table. If you don't draw a table, you lose a mark. And then we say that make sure that you tell us that blood group A is complete dominance, while blood group B, we say that blood group B is co-dominance. We talked about that. We said that blood group A, yes, it has one recessive allele and one dominant allele, while blood group B, both of them are equally dominant. And then we say that if you can't remember that, you can even say they have these alleles, while this one has allele for that and allele for that. So then you can also talk about that only dominant allele is being expressed in the what in the blood group A, because it is complete dominance, while the other side is both alleles are being expressed in the phenotype of an organism. All right, so we're here, and then we say that use the genetic equals to show the parent with with at least 2 blood groups in the world could have children with the blood group A. So they're saying use the least common. So we said that the least common is blood group AB and blood group B, because if I add them, I'm gonna have this one is 16, or if I add here, I'm gonna have 5.5. So it looks that blood group B and blood group AB are both low in numbers. So they're saying that so I'm gonna say P1, yes, is P1. I'm gonna say P1 F1. I said F1, I'm writing P1. All right, F1. Then from there, I'm gonna come back and say that what is the phenotype? What is the phenotype? So we see that the phenotype of this is blood group A crossed with blood group B. Don't just say A. Is it B? Blood group AB, isn't it? Because we say it is blood group AB. Blood group A and blood group are they the one with, yes, blood group A and blood group AB. Yes, blood group AB. All right, then I go to genotype. My handwriting is ugly. Genotype. Then I'm gonna say the genotype of blood group A, it is A. Why am I saying blood group A? You see. It's not blood group A, it's blood group B. You guys, you must tell me in the chat that is not blood group B. Blood group A. Blood group B. Okay, this is blood group B. And this is blood group. So the genotype is blood group B, but they want to produce blood group A. So it means that this blood group B, it can't be blood group like heterozygous, I mean homozygous, because if it is homozygous and then crossed with the A and B, so this will combine with one of this and then it's going to produce A, B. You understand? So it means that this blood group B, because we want to produce a kid who is blood group A, this blood group B must be, because we want to produce this kid who is blood group A, this must be heterozygous. You understand? So so that is supposed to be heterozygous. So now it means that when I go to my meiosis is supposed to give me B, and then it's supposed to give me that, and then this one is supposed to be give me A, and it's supposed to be B, like that. And then I say, how do I call this? We call these ones gametes. And then after that fertilization, I told you you can draw a Punnett square, or you can draw 4 dots, so that it's easier for you to write them down. Someone said, genetic cross, I used X instead of I. No, this is not sex linked. They didn't talk about sex linked, guys. I always tell you that if they don't talk about sex linked, please don't use don't use X and Y. Yes. So this one goes, you join that bullet with that, so it's gonna be AB, and then this is gonna join that with that, so it's gonna be why did I say yes? It's gonna be B, and then now this is gonna go with that, is gonna be A, heterozygous, and then this is gonna go with that, so it's gonna be B, heterozygous. So do I have any chance of having blood group B, of having blood group A? So these are offsprings. Cool. So now I come here and then I find out what is the phenotype and what is the genotype. So basically the genotype here, I have blood group A, B, I have blood group B, homozygous, and then I also have blood group A, heterozygous, blood group B, heterozygous. And then the phenotype, I'm gonna have blood group AB is 1, blood group A, B, blood group, don't just say blood AB. And then I'm gonna have blood group 1, blood group B, and then I'm gonna have this and this are blood group B. So I have 2 of them, and then one blood group A. So that's what's supposed to be written in that regard. Yes. Did you get it right? Let me see, what are they saying? They say the parent with the common blood group. I use the AB for both parents. They say the parents with the common blood group. They say the parent with the common blood group. Yes. So when they say someone is saying that parent with this let me explain to you, maybe you didn't get it. They say that they say here that use the genetic cross to show at least 2 with the to show how parents with the 2 least common blood group. Remember, you have the blood group AB, which is positive and negative. You understand? So this is the same blood group, common blood group. This is also this. So if you add this and this common blood group, you get a figure. If you add this and this, you get a figure. If you add this and this, you get a figure, and you get this and this, you get a figure. So the lowest is the one which they were asking. 2 of them. So that's what they are saying. Because if you say this is lowest, this is low, this is low, this is low, this is low compared to these black ones. That's why they say the common blood group, so that when you combine them, you get it. All right, we continue to the next question. They're saying that 2.5, that the CCR5 gene in human is located on chromosome number 3. This gene is responsible for producing a protein on white blood cell. The HIV normally uses the protein to attach to infect white blood cell. A mutation of CCR5 gene appears to offer some protection against HIV by changing the shape of the protein on the white blood cells. Scientists have discovered that the long-term reduction of HIV in the patients is possible through the stem cell transplant from the individual with this mutation. Bone marrow is rich source of adult stem cells, the stem cells can develop into a new white blood cell. Name the type of chromosome with the CCR5 gene found. Name the type of gene, name the type of chromosome on which CCR5 gene is found. Chromosome number 3. So that is orosome. So that's autosomal chromosome. So automatically I'm gonna say that, because if it was 22, then it would have been 23, it would have been sex chromosome, but they're saying chromosome number 3, so it is autosomal. Yes, autosomal chromosome. Define stem cells. We talked about. I remember when someone said, will they bring stem cell? Why are we defining it? Say define it. Okay, basically is undifferentiated cell which have ability to divide and develop into other specialized cells. So that's how you can define it, or any other, but you have to show us that stem cells are undifferentiated or unspecialized. You can say unspecialized, undifferentiated. They have ability to divide and grow into other, or develop into other any type of tissue in the body. Yes. Then they're saying that use the information provided, explain how stem cells can, from the person with the mutation, could reduce HIV virus in the patient. They say that some discovered that long-term reduction of a patient is possible for stem cells for an individual mutation. Bone marrow is stem cell, develop into a new white blood cells. Okay, we see that they also talked about it here. Mutation of gene offers against the changing the shape. You see, it changes the shape. Normally uses the protein attached. So we see that first of all, the stem cells can develop into new white blood cells. You see, into new white blood cells. These stem cells can develop into new white blood cells. Then number 2, the proteins can they can develop proteins that have different shape. Yes, white blood cells, they can develop proteins which have different different shape. So it means that they cannot be recognized. The HIV cannot recognize it. They cannot recognize this HIV, and then it means that it will not be able to infect the person. Why? Because it is a different protein. It's like me, I give you a key, and then when I give you a key, to open at home, you end up not opening a different key. You understand? I give you a different key other than the one which you have, the one which you used to open with. So it is a different one. So it means that you cannot open. So you have changed the protein. This HIV is supposed to attach itself. You understand? So once you change, then automatically the HIV won't be able to detect these cells, these white blood cells, and then the white blood cells will be safe, and then you'll be able to survive. Yes. That is stem cells. And then we go to question number 3. The last question, they're saying that which syndrome is this? ASA is a rare genetic disorder. Yeah, actually it's rare, caused by recessive allele. So they talked about it here. Recessive allele, X chromosome, that is sex linked. So whatever you are doing, you must have X and Y. Located on X chromosome, the dominant allele X result in the normal physical feature. The people with ASA often have the following features: wide spread of eye, okay, small nose, long area between the nose, okay, wide peak hairline, okay, squat, and descended test, okay. They're saying that if you're male, may God forbid, those things don't happen to us. Okay, we are saying that you have the pedigree here. Use the information provided. Okay, use the information provided. State 2 characteristics of ASA that are unique to males. So they're saying unique to male. Oh, unique to males. Automatically they said this. So you talk about that, and they also talked about scrotum. You don't have female who have scrotum. No. Yeah, so these are unique to males. All right, then we go to the next question. They are saying that define the recessive allele. A recessive allele is an allele which is being masked in a heterozygous state. You understand? It's being masked, or this is an allele which expresses itself, you understand, in the phenotype when only in a homozygous state. It's the one which expresses itself in the phenotype of an organism when only found in a homozygous state. You understand? Or is the an allele which is being masked by a dominant allele in a heterozygous state. Yes, so that is it. How many offsprings are individual 8 and 9? We go back and look for individual 8 and 9. We see it's 1, 2, 3. This one just got married to them. So the answer is 3. I hope, guys, you are getting it right, even if you don't get it. I know that there are some people from different provinces, they are using this as a guideline, as a revision, and other people for other papers, final exams. You can use them and then you understand. You are not just here to see your marks. I know that you are here to see that if they bring the same question or similar questions, you're able to answer that. That's why I try to explain this. Yes. 3.1.4 state the phenotype of individual 11. Individual 11, we are seeing that is a male with what? With the ASA. Don't say is affected male. Affected with what? HIV? Affected with what? Malaria? Affected with what? What is it affected with? So you have to tell us affected with ASA. Then they are saying that genotype of individual 4. They are looking for the genotype of individual 4. Individual 4 is a female. So let me first put individual is a female, but this individual is normal, meaning that he has what is the capital letter is A. But now we don't know whether it's homozygous or is heterozygous, but they produce a kid who is affected with the ASA. You understand? So it means that you're gonna say this came from the father, then automatically this came from the mother. So it means that individual 4 is heterozygous. So it means that individual 4, I come here and I say that individual 4 is A and individual A is yes. Something like that. So, Zoo also, you are seeing Max, unfortunately, Max are going where? Anyway, meaning that is basically seeing miracles in the paper. Okay, explain why female are less likely to inherit ASA. Remember, this is they're asking for female, they're not asking for male. Females have 2 X chromosome. That is a first mark. They require 2 recessive allele, yes, for them to be having ASA condition. You understand? If one recessive allele appears on X, it can be masked by a dominant allele on the other X. So the individual is still what? Normal. Yes. So that's what you're supposed to explain or link to it. Yeah. Then we go to the next point, but it's supposed to be linked to that. You have to show us that the females have 2 X, you see, and then they have to be with the, you can be like this, or you can be like, for them to be affected with this condition, ASA condition, yes, you have to be with the 2 recessive alleles. If one recessive allele appear on this X, it can be masked by this dominant allele, and then the individual is still what? Normal or is free from ASA. Then we go into question 3.2. Cloning is a type of biotechnology used in the by people who want to keep their beloved dog in their lives. It always it allows them to produce genetically identical dogs to their previous ones. However, it is very expensive process and reduces genetic variation. Expensive and reduces genetic variation. During the process, nucleus from the skin of the dog being cloned is removed to be placed into the ovum. You see, skin, ovum, this one is 1N. While skin of the dog, it is orosoma. This is 2N. Huh? Yes, you need to know that. As I explained it before you went to the exam. Once the embryo of the clone dog has formed its genetic implant into the uterus of the surrogate female, dog, where it will develop. Surrogate, remember, is the one which helps to develop the embryo. Further until the clone dog is born. All right. Based on information, state 2 disadvantages of cloning. We see it's expensive and reduces genetic variation. Then they're saying that explain why the nucleus of the ovum was replaced with that of the skin. Remember, the ovum has one end, as we have said it, while the one which is the skin is 2N. So we need this nucleus to be inserted into this so that this cell looks like a zygote. Number 2, we did not want the characteristic of this individual, of this nucleus. So the nucleus of the ovum has no characteristic which we are interested in. The one which has the characteristic is the nucleus of the what? The characteristics of from the skin. So basically, when the nucleus of the skin is inserted into the ovum, it creates the diploid state. So that's what we are talking about. And then since it creates the diploid state, and this nucleus, this one, it has the desirable characteristics we are looking for. You understand? Then the nucleus of the ovum is haploid. We know that. Therefore, it has basically, it does not have the complete set of the genetic material which is needed for the dog to be produced. Basically, it doesn't have the characteristics we are looking for. Yes. So that's why we have to throw away the nucleus of the ovum. Remember, I don't want to repeat the process here because I explained very well. I've created a number of videos which are trying to explain this, even in using our distinction material book. And I didn't want to talk about it because the people who use it, they know the benefit of it. And they know that, yes. By the way, if this is your first time to visit our channel, don't forget to subscribe, like, and share. Always tell them that the best channel is here. All right, we continue. We got that the frogs produce different sounds to attract female. Well, last time when I was explaining this situation, someone was even saying that, did I hear that? So you heard it. I hope you have written it. Yes, when I was differentiating for you the different sounds. So they're saying that frogs produce different sounds to attract females of the same species to breed side to the breeding side. They are saying that the call signal that start of a breeding season. Okay, this breeding is in summer, and then the male makes strong. All right, chorus sound. All right, it means they're singing, eh? All right. However, this breeds in springs and male makes deep, low pitched whoops. Okay. List 3 requirements that organism must satisfy before they can be called species. Remember that a species, they must be similar organisms. They must be able to interbreed to produce a fertile offspring. They must be able to interbreed. They must be similar organisms. They must be able to produce fertile offspring. Guys, I've been telling you that we don't accept viable offspring because it means something totally different. So it must be fertile. They're able to produce fertile offspring, must be similar. They must be able to interbreed. So that is a species. Use the text above, identify and describe 2 reproductive isolation mechanisms these frogs have. So basically, you will see that they have different way in which they dance. So that is species-specific courtship behavior. Those people who were watching my video previously, I think you know what I'm talking about. That's what I was talking about. Species-specific courtship behavior. And I say that each species have a specific way in which it does its courtship. So that behavior belongs to that specific species. And I said the example of these frogs I gave, and I said that if the frog is doing when it is, you don't like it, you don't want it, but for the frogs they are enjoying it, and then that's why the reproduction is still continuing. Yes, so that is one of the species specific courtship behavior. Then also they are breeding at different times of the year. We see this one is in spring, and then the other one they said is in summer. So you see that breeding at different types of what? So how many marks? So since these 4 marks, so if I say species specific courtship behavior, will I be able to get marks? No. So you tell us that species, you can mention the species which are being involved. You mention the species, and then also when you talk about the different time of the year, you tell us which one is in summer, which species in summer, and which one is in spring. So you are able to get maybe at least those 4 marks. So it will depend on the memorandum how they have done it, and then yeah. Maya, I'm seeing Speciation. You guys, are you? All right, we continue the next question. Frog become reproductively isolated because they are different species. This may have happened due to geographic isolation. Yes, describe the process of geographic isolation. Here, they are not asking you about, they have not given you any example, so I'm just use whatever I'm using. Remember, we said that if a population, or there was one population of a single species, this population is separated by a geographical barrier, or you can say separated by a river or what? Yes, by geographical barrier. The population, the two population or subpopulation, there is no gene flow between these two populations or subpopulation, whatever. I want to see the word population. Please don't mention about species, because here the species have not yet formed. It's still one species. So the population are split into two populations. There is no gene flow. You understand? And then they will experience different environmental conditions. Therefore, natural selection occurred independently. You understand? Then they become genotypically and phenotypically different. Even if you remove this barrier or this whatever barrier you have used, they cannot mix, they cannot interbreed to produce a fertile offspring. They cannot interbreed to produce a fertile offspring. Since they said here that they have already formed new species, I don't think they're going to award that mark. Yes. Yes. By the way, let me ask you a question. Let me know, guys, which schools are you coming from? Let me tell me in the comment section. Yeah, at least I need to know the people I'm basically talking to whenever we are doing these live classes. Yeah, I just see specifically Mbata, I just see George, CP, I just see people. I don't know where you are coming from. Let me know. Pindile, Sikosana, I see you. Zoe, I see you. Fortunate, I see you. Let me know which school are you coming from, so that at least I'll recognize the school. Sometimes we have gifts, but we don't know whom should we give, because we don't know where are you coming from, basically. All right, let's continue. Hey, Lambano, congratulations. I didn't know, guys. Tulani Secondary School, Lambano, Mosi High School. All right, okay. Thank you so much, guys, for being great. Yeah, I hope you guys are benefiting from this channel, so that we are here to serve the country. Oh, wow. Whitestone High School, Maharang Secondary School. Guys, let me continue with the discussion. You're just making me happy. Just making me happy. Yeah, I'll take a screenshot of that, those schools, so that at least maybe I can come and also do a physical class there direct. All right, tuberculosis, that is caused by an infection by a bacteria called Mycobacterium tuberculosis, can be a life-threatening. Resistant strain of Mycobacterium tuberculosis have evolved because of patients do not complete their antibiotics. Guys, make sure that you take your medication properly. I don't know, it's okay, this is one of the antibiotics used for a first line treatment for TB, to prevent TB from becoming active. By the way, did you know that all of us, we have TB? Yes, we have TB in latent stage, TB in inactive stage. But if your immune system goes down, it means that TB is being exposed, meaning that it starts to show up. Yes, so that's why TB is very common in people who are having immune deficiency. Yes. All right. A survey conducted by the National Institute of Communicable Diseases from 2013 to 2014 compared the percentage of patients in whom TB has evolved resistance in these antibiotics with the 2001 2000 survey. Okay, meaning that these people, they were just reading some articles which were done, and then they are trying to do the summary. All right, they're saying that the survey was conducted as follows: 349 patients with tuberculosis received treatment with ionized. Okay, selected, they were from South African hospital or clinic, all 9 provinces. All the patients were over 18 years, and then the patient medical records were reviewed. All right, Naledi High School. Oh, guys, thank you for being here. All right, they're saying that the table below shows the percentage patient in TB has developed resistance ionization antibiotics of 9 provinces for both surveys. All right, the province, we have different provinces. All right, name the theory that Charles Darwin proposed to explain how Mycobacterium tuberculosis is evolved to become antibiotic resistant. So we call this is a theory of natural selection. Yes. We call it Darwinism because it's Darwin who did this theory. But this Darwin explained different concepts. In this case, we have to be specific. Which concept are they talking about? That is the theory of natural selection. That's what they are looking for. Then they're saying that identify the dependent variable. What is the dependent variable? Here we see that the percentage of patients in whom TB has developed resistance. You see, it means that developed, basically here we are saying development of resistance to this antibiotic. You understand? Yes, development of resistance to this antibiotic. Someone is gonna say, me, I wrote this. Maybe they will give you a mark for that, but I'm not so sure. But this question, whenever they bring these questions of antibiotics, you have to read it properly. You remember this in grade 11 when they are bringing this SBA. They still brought it, and then it was a bit challenging. Which province had a highest percentage of TB patient with the resistance between 2013 and 2014 survey? We see this one. I see it's this one. It's Northwest. Northern Cape. Then they're saying that state one way in which the reliability of this investigation was. So it means that you have to talk about how did they ensure reliability for this investigation. How did they ensure? They basically ensured patients were over 18 years. All patients were from the same area, that is South Africa. And then you can talk about same duration of the survey. It is basically they did for 2 years. You see, yes, they did for 2 years. All patients had the same disease. There is no one who was not having the disease, so all of them were affected with TB. So and others which you could give. Then they are saying that calculate, what are they saying? Calculate the average percentage of patient who had the drug resistance to South Africa during 2013-2014. Okay, so it means that it's, let me see, 1, 2, 3, 4, 5, 6, 7, 8, 9. So you're gonna have to add all of them. After adding all of them, you divide by 9. So all of them divide by 9, and then you are able to get the what? The marks. And then they said, oh, there is something which I didn't see. Round off to your 2 decimal places. It means that if it is like the answer you get, it must be point, yes, point, maybe 3, 4. It must be. If it has a lot, you have to cut it to 2 decimal places. Yes, someone is saying 5.36. If it is 5.36, yes, I don't have a calculator here. It is 5.3636. So it means that is it supposed to be percent. Then they will give you a mark. But if you are not having this decimal, because here they said 2, so they might cross it. I'm not so sure, but they might cross it. Draw the bar graph. You know, you have to know about the bar graph. And they are saying bar graph representing 2013-2014 survey results of Eastern Cape, Free State, Northern Cape, Western Cape, Northwest, and then Northern Cape. 1, 2, 3, 4. So it means that if you're having here, they are looking for only 4 of them. So you had to scoop out only those 4, and you draw a proper bar graph, because there we're gonna have to mark for the title. We see that the title is correct. We're gonna have to mark the type of graph. That's why you see that they say title, that is the caption. They give you a mark, at least 1 mark. The type of graph, have you drawn a bar graph? Whether it's correct or wrong or what, as long as it is a bar graph, we give you a mark. Then we're gonna mark for you the labels. The labels must have units. Yes, this and this must have units. So I will say that the labels will give you a mark. And then we're gonna mark the scale. Do you have proper the ranges, that 2, 4, 6, 8, 10? Yes. So if you have, also we give you the mark for that scale. And then we're gonna give you a mark either 2 if you plot all of them correct, or 1 if you only plot 1 correct. What about who drew all of them? That's where you might lose a mark. If you might lose a what? A mark. Someone, I divided the number by patient. Sorry. So we go to the second last question. They're saying that a map below represents the hypothesis used in the evolution to describe the origin of modern human based on the age of fossils discovered worldwide. Okay, identify the hypothesis represented by the map above. So describe how genetic evidence identify the hypothesis representing by the, what hypothesis is this? It's out of Africa hypothesis. We see that we originated here, and then you started to move other parts of the continent, of other parts of the world. That is out of Africa hypothesis. They're saying describe how the genetic evidence can be. Describe how genetic evidence can support the hypothesis identified in the question above. That is genetic evidence. They're saying describe how the genetic evidence. Remember, when you talk about, there are 2 evidences which you're supposed to talk about here. That is the genetic evidence, and then that is the fossil evidence. Yes. So based on examination guideline, now, please teachers, you need to know that the only evidence for supporting the out of Africa hypothesis is genetic evidence and the fossil evidence. But if you are describing the out of Africa, you are describing the fossil, then you talk about if you are describing the originality of the species, then you describe all of them. you describe the genetic evidence, the cultural evidence, and then the genetic evidence, the cultural evidence, and then you describe what? You describe the fossil evidence. But if you are explaining the out of Africa hypothesis, is only 2 of them, that is fossil evidence and the mitochondrial, not mitochondrial, the genetic evidence, which explains the mitochondrial DNA. So mitochondrial DNA is passed from basically mother to a child. Yes. So when you study them through studying of the mutation in the mitochondrial DNA, yes, female line ancestry can be traced. Yes, that the oldest is found in Africa, which shows that modern humans originated from Africa. Yes. So that's how you're supposed to explain. So you explain the mitochondrial DNA. Name one type of evidence used to support hypothesis identified above, other than genetic. So it is fossil evidence. That's it. No cultural evidence. Yes, it is fossil evidence. Why? Because based on our examination guideline, that is supposed to be fossil evidence, and then genetic evidence. Cultural evidence, no. Yes, cultural evidence was there in the previous examination guidelines, but the current examination guideline we are using, it has no cultural evidence when you're describing the out of Africa hypothesis. The paper is done, Yeah, guys, thank you so much. I'm so happy to see that you guys, you have been with us from the beginning until the end. I see that people from Pretoria, I see you guys. Okay, I see you guys. Oh my God, I see you, a lot of people from different schools. I want to know, hello. Oh, I wanted to know where is specific Butelezi is coming from. I wanted to know also, there is this one who is always talking a lot. Who is this? Who? Sipesise Mbata. I don't know some of these schools, but I'm gonna search them on the internet to find out where. But guys, thank you so much for being with us. It has been a wonderful. I think this paper was fair, and at least when I analyze this paper, it looks like the KZN paper. And if you look at our prediction, guys, our prediction based on our revision, we have killed it. I've not seen any paper there question which is, I see at least 98% of our revision we have tried to smash this paper. I don't think there is anybody who has been following us and the paper smashed him or her. I know that you have smashed the paper. Thank you so much. See you again, guys. For finals, we have to start preparing for finals. We must start now, and then we prepare for finals. Those people who are joining us to the camp, the study camp, yes, we are waiting for you. You can check upon us on WhatsApp, and then those who are joining us for the study camp, yes, for 4 days we're gonna be smashing all the subjects. Yes, all the subjects we're gonna have to prepare them. And our motto for that time or slogan, everyone must pass. And I mean it, everyone must pass. Yeah, thank you so much. It is Msaidi. See you again in the next or in the upcoming preparations.